Hi, I hope everyone's having a great day. This is for biology. We are going to be working on Unit 7, Genetics, Notes 4, which is over sex-linked traits and pedigrees. Today we will be focusing on learning how to predict genotypic and phenotypic ratios of sex-linked traits. And we're also going to learn how to track the passage of a trait through a pedigree. So, we're going to focus on looking at what a sex-linked trait is. Some traits or genes are only going to be located on the sex chromosomes. If we think back to the karyotypes that we looked at for humans, humans have 23 pairs of chromosomes. 22 pairs are just normal chromosomes, and that final pair, the 23rd pair, is what determines the sex of an individual. An XX is going to be a female, and an XY is going to be a male. So when a specific trait is located on these chromosomes, it is considered to be a sex-linked trait. And basically, to sum that up, it's a trait whose inheritance is related to the sex of an individual. Some examples include muscular dystrophy, hemophilia, and color blindness. So here we see an image of somebody who is a carrier for color blindness and a male who has normal vision. So let's bring that up here a little bit. And what we are going to look at now is here we see a female. She is considered a carrier. Carrier means that you have one of the recessive alleles here. So here we're calling it a defective allele. It's typically a recessive allele, not always, but most of the time is recessive, and a normal allele. So to be considered a carrier, you don't show any symptoms of what you are looking at. So in this case, this person has normal vision, but they carry the allele for color blindness. Over here, you'll notice the male has a normal allele, which is gray, and the Y chromosome. And you'll notice there's no allele for this on the Y chromosome. That is what makes it sex-linked. And you'll notice there's some interesting things that happen. So once again, mom and dad both can pass on either of their alleles. So for this first possible offspring, we have two gray normal alleles here, which means she got an allele from mom for normal and an allele from dad for normal. So you can tell XX, female, normal, normal, normal vision. Looking at the second individual here, we have another female, XX. She has a defective allele, and so looking at the parents, we know this defective allele came from mom, and then that means the normal allele came from dad. She is also going to be a carrier, which means she has one of the defective alleles but is not showing the trait herself. And now as we come over to the first male child, we have a normal allele. We know the X had to come from mom, so mom gave her normal allele and dad gave his Y. Normal vision, nothing really going on there. And now we come to this final child. Once again, another male, XY. He got the defective allele from mom and the Y from dad. So what happens with this child is he will be colorblind. That is because he only has that one defective allele. And with no other allele to balance it out, he's going to show this colorblind trait. So now we are going to look at a possible cross. We are going to cross a male right here who is colorblind. So we're going to represent that by using an X with a little n and a Y. And then we have a woman who is a carrier for the colorblind trait, which means she has two X's, and her superscripts up here are going to be a big N for normal blindness and a little n for colorblind trait. So this woman, though she is a carrier, remember she herself would not be colorblind. So we're going to look at this Punnett square here, and then we're going to look at the genotypic and phenotypic ratios. So we're going to start by putting in their information. So we have X, and I'm just going to put a big N because trying to get a superscript on here is a little bit of a pain. And then we're going to go over here and finish up with mom, and we're going to do X little n. We are going to come over here and put dad, and dad is X little n and a Y. So this is a normal box. We're going to go ahead and fill it out. So we're going to do X big N, remember that is coming down from up here, and the X little n coming from over here. So remember the X's just tell us it's female, and when we're looking to see if they're colorblind or not, we're going to look at the N's. So let's go ahead and come over to this one. Once again, we're going to bring it down from the top. We have X little n, and bringing it over from the left, X little n. Third box, bringing it down from the top, X big N, and Y. And then over here, bringing it down from the top, and then from the left, we have X little n and Y. So when we look at these, we know we have two females. Obviously the top are both female, and the bottom two are both male, XY, XY, XX, XX. So we're going to come over here and do our genotypic ratio. So when we look at this, all four of these are different, so it's going to be pretty easy to do. So we would have one X big N, X little n, then we're going to do our colon, and then we're going to have one X little n, then a colon, and then we're going to have another one X big N, and then Y. And then finally, our last one is going to be X little n, Y. So this looks like a bunch of confusing letters, right? Even with the superscripts, it would still be somewhat confusing. So we're actually going to take this genotype ratio up here, and we're going to put it into a phenotype ratio. So what would we physically be seeing out of this matching? So with them all four being different, and we look at them, they all four actually code for different things. So the first one is going to be a female who is a carrier for colorblindness. Next, we are going to have a female who is actually colorblind. Two little n's would be colorblind. Next, we're going to start with our men. We have one male who is just normal. Men can't be carriers, because to be a carrier you have to have a big and a little, and they can only either have big or little. So you will never have a male carrier for colorblindness, at least. Any sex-linked trait, you can't have a male carrier. And then finally, our last one is one male who is colorblind. So once again, our genotype ratio is up here, and this is going to be our phenotypic ratio, telling us basically what we would physically see. So what we would actually see, even though we know she is a carrier, this female would only show normal vision. And we know with this cross, any female is going to be a carrier if they are not colorblind. Because to be female, you have to get your X from your dad, and the only X he can pass on is a colorblind, which means every female is going to be at least a carrier, if not colorblind themselves. If you wanted to simplify this down a little bit, we could do 2 normal to 2 colorblind. Because when we look at the offspring, we have 2 that would come out normal and have normal vision, and then we would have 2 that are colorblind. So moving on to the next one, we're going to look at hemophilia. Hemophilia is a blood disorder, and typically with hemophilia what happens is your blood doesn't clot, so if you get just like a simple cut, it's a lot easier to bleed out. So with this one, it doesn't give us letters, so we get to pick our own. So we are going to go with, once again, we're going to use the X and the Y in addition to another letter. And so for this letter, I'm just going to use H for hemophilia. Once again, you can pick any letter. I recommend if you are handwriting these to pick a letter that's significantly different from the uppercase and lowercase. So we have a normal male and a female carrier. So we're going to start putting the mom up on top. So she is a carrier, so that means she has the X, she has a big H, and then she will also have let me go ahead and move that up a little bit. She will have X and little h. So remember, big H, little h would be a carrier. Carriers always have that heterozygous genotype. We have a normal male. So let's come over here and do our normal male. He's once again going to have an X with a big H, and then being male, having a Y. So let's go ahead and fill out the boxes. X big H, X big H. Coming over here, X big H from the left, X little h from above. Once again, convention just says that we write the big letter before the small letter. Down here, we are going to have X big H and a Y. And then over in this one, finally, we are going to have X little h and a Y. So when we look at these, we can see that all of these, this one, and this one, all have that big H. With that big H, that means they are going to not have this disorder. So we have 3 that are going to be normal, and then we have this one male child here that is going to that will have this disorder. Now, if this couple were to have a female child, they would not have to worry about that female child having hemophilia. They would really only need to worry if they have a male child, and then they would need to make sure to test whether or not that child has hemophilia. So if we want to look at this percentage-wise, we have 3 normal and 1 hemophilia. And let me look at how to spell that because I know I will misspell it. 1 with hemophilia. So 3 normal, 1 hemophilia. If we do percentages, we would have 75% normal and 25% hemophilia. So their chances of having a child who will have this disorder are 1 out of 4. So 1 out of 4, 25%. So those are sex-linked traits. They work identical to all of what we've been doing already. So moving on to pedigrees. When you hear the term pedigree, the first thing I think of are dogs, because a lot of, especially show dogs, they talk about how good their pedigree is, which is referring to their family and what their family line looks like. You also may hear about it in terms of racehorses or things like that. So to simplify it down, a pedigree is just going to be a family tree, and it's used to track the passage of a trait. So males are going to be represented by squares and females are going to be circles. Pretty straightforward. So when we look at this, we have a male and a female. So the male and the female, when we look at the line down, this is going to be generation 2. They had 3 children. This one is a female, this one is a male, this one is a male. This male met another female and produced 3 offspring. This female met a male and produced 3 offspring as well. So remember, you're just tracing these lines where they go. We're going to talk about what the colors mean here in just a minute. So we're going to label all the infected individuals first. So we are going to do that by marking them with a line, and let's go ahead and make that a slightly different color so that it's a bit easier to see. So the black ones are going to be the ones who are infected. So here we have one individual who's affected, affected, affected. Next we are going to mark, and we are going to use blue for this one, all of the normal males. So remember, males are squares. So here's one normal, here's another normal, and here's our final normal. Then we are going to label all of the normal and carrier females. So remember, she's showing a trait. If they are dark, they are showing that they are the infected trait. So what we look at is this one shows an infected trait, this one is not infected. We know that to get this infected trait, you have to have one trait from each parent to show it. So these individuals both got the affected traits. This individual we know had to get one affected trait and one non-affected trait. So remember, because she's heterozygous, she has one of each, she is going to be showing that she is actually going to be a carrier. So carrier. And then down here, we know that both of them are carriers because they produced one child that had the infected trait, or the affected trait that we're looking at. What we don't know for either of these in particular is whether or not they are carriers. But we also know that she would be either normal or a carrier, so we're going to go ahead and mark her as well.